Dear friends and colleagues, it's a big honor for me to have the keynote speech here at this Congress. And I struggle a little bit to find the right start, since being forced myself more than 30 years ago to serve for 1.5 years in what was this time basically an outlet of the Russian army, I at least have a rough imagination what war means and how it feels lying in a trench with a rifle in front of you and not knowing what will happen in the next minutes. Of course, that was all training, that was all peace, but at least I have a little bit feeling. Even more, I'm happy that we are here in science, and science has, on top of all other functions, one very important social function, and this is connecting people. And therefore, in this talk, you see I have just added the country codes to numerous persons whom I owe thanks, which contributed to this talk, and which all serve with us to unite science. Since this is maybe the privilege of science, we have to think a little bit ahead. Russians and Ukrainians are neighbors by destiny. There is nothing what can change that right now. So we have to think also for the time after the war, as cruisome as it sounds right now. Therefore, I'm super grateful first for our Estonian hosts to have hosted this Congress. And especially grateful I am, and now comes the other flag, hopefully it will work. No, let me see. Yes, it does. To our Ukrainian team, namely Irina, Dimitri, and Anastasia, which worked diligently to have this Congress up and going. I will a little bit talk about what are the challenges for systematics and ecology of myxomycetes from my point of view, since this enigmatic group of organisms is nowadays treated as rarely another group to be researched together with institutional and non-institutional scientists, as I would prefer to call it. Of course, we have also names like professional amateurs or citizen science, but we are all citizens and we are all professionals. So institutional and non-institutional may also be okay. And now I look here at the screen. I want to divide my talk in 5 parts. Myxomycetes, let's first have a small look about their main evolutionary tendencies, which explains a lot of the difficulties we will face in further research. Second, the dimensions of Myxomycete diversity. I call them visible, cryptic, and hidden diversity. Third, what should we do, or what can we do for fruit to come to a really, and I think Angela has coined that word, integrative taxonomy, just more than only fruit body morphology, although fruit body morphology is super important as well. Then the quest for the natural system in myxomycetes and its consequences. What difficulties we will face with exploring into that. And second, a little bit the ecology, where I think we have one major gap in myxomycete ecology, which is the mostly unknown link to the food organisms right now. And I will just divide my talk in 3 kinds of slides. Let me see if the pointer works. It does. There's a little red dot first. The green is our achievement. The red is our challenges. And then I have brought you a little example of a yet undescribed species in myxomycetes we tentatively call Diachiopsis resinae, and it shows very nicely the difficulties we face in just doing myxomycete research. So I will always go back to this species as an example, and you are of course invited after to have your critics and suggestion what we could improve. So myxomycetes are one of numerous or several protist groups just evolving at least towards macroscopic size. I'm a little bit hesitant, I'm a little bit reluctant to say multicellularity, since true multicellularity, which means numerous cells growing together in an organized way and have a crosstalk, that's still another thing. Some thoughts about the evolutionary significance of the fruit body structures. And here you see a wonderful drawing of my friend Dimitro. He knows it. He is not only a researcher, he's also an artist. And he has drawn this still simplified, in spite of all the many icons, still simplified tree of the eukaryotic life. And you see the myxomycetes as one of many groups. Let me point to these. I always have difficulties to find the red dot. Here it is, and the red dot goes along proderma, which is right beside the epithelium. These things you can recognize. Many others, probably only the specialists of respective groups, can recognize, since these are numerous other eukaryotic groups. And when you look at these groups, many of them are protists, living more or less predatory, being more or less mobile, and of course, basically being unicellular, kind of unicellular animals, to say so. But our myxomycetes later on, and that makes the interesting thing of the myxomycete biology, they are first animals, and later they become, let's say, fungi when it comes to their dispersal ecology. And we all know that's the reason why traditionally mycologists have a little bit always looked at myxomycetes. There was a nice contemporary review just reviewing the numerous ways to multicellularity. The approach of the myxomycetes, as you know, is the plasmodium, but also the plasmodium is not unique. There are several more groups which have plasmodia, and when you look at those which are forming fructifications, that is also not a unique achievement of the myxomycetes. With my dot, I have problems. Fructifications. Here, of course, the true fungi have kind of fruit bodies. So you see numerous groups which kind of form fructifications. This little number 65 points to the unique ciliate Sorogena stoyanovice, which forms fructification. I've seen it once in moist chambers in several colonies. It looks like an overdimensioned echinostelium. Later on, who wants to know what all these groups are in the PDF of the talk? The interesting thing here is, and that is our problem, I think, in myxomycetes. I will come back to that numerous times. In the Amelozoa, numerous groups have just developed spores and fruit bodies and have been evolving towards macroscopic size. You see this in this diagram, which I show usually to the students, since you have different, let's say, levels of observation from the naked eye over the hand lens, over dissecting microscope, a compound microscope, back to the electron or scanning electron microscopy, and you see these little peaks here that several of these myxomycete groups here, the Acracetes, Copromyxa, the Protostelites, the Dictyostelites, the Ceratiomycetes, and of course the myxomycetes evolved towards macroscopic size. There's one other interesting exception, which is not evolving to macroscopic size by spores, but by a shell. They look like miniature snails, the Arcelimita, the shelled or testate amoebae. So these groups, although more or less independently and acquiring these adaptations independently, we can name as a kind of ecological guild myxomycete and myxomycete-like organisms. And if you project this on a phylogeny, of the Myxomycete, and this is only one of the big subgroups, Terosa, then you see the fruit body, or let's say more cautiously, spore formation. That spore formation has appeared several times. All the groups which have a black dot here in the tree, and that is the tree of this wonderful paper of Kang et al. 2017, a phylogeny of the Myxomycete, you see numerous groups have spores. First, the evolutionary idea is obvious and very easy. The cell converts itself into a spore and forms secretes a more or less long stalk. That's what we call the primary stalk in Myxomycetes. And usually then we have only one spore. So the red numbers at the side of this phylogenetic tree depict the number of spores, and you have usually only one spore. But then in the crown plates of this group, the so-called Myxomycete, you have groups which have numerous spores. I would even say the spore number kind of explodes at the end. And this is where we are, this red triangle just at the bottom of this slide. These are the Myxomycetes. So this is a striking evidence that spore formation is a very common thing in living organisms if you want to go out of the sea. And if you can't fly or can't run over large distances, and maybe are not so good runners, of course, you need to develop a propagules, you need to develop spores, and then a whole number of evolutionary constraints immediately kicks in. These are, you have to be small, you have to be more or less round, and you have to develop a resistant shell, a spore wall, to be a little bit sheltered from desiccation, but also from ultraviolet radiation. So, and then on the end, to get these spores really airborne, you need to develop a stalk. A stalk is essential, as we will see in the next slide, and that stalk here has also developed several times. And what is even more interesting, even the prokaryotes can do that. And when we look at evolutionary tendency, it's really nice to see that the first group are the myxobacteria. They develop a stalk. If the students ask me, can you see bacteria? I say, yes, sometimes you can see them with the naked eye. On the flowers of big tropical plants like Ecology or Psychotria, or these large conspicuous flowers, after a good rain, the myxobacteria will bloom and you see them with the naked eye. So the stalk is as well a product of parallel evolution. Different groups have had different approaches to develop a stalk, and as you will see, even in the Myxomycetes, there have been different approaches to develop a stalk. And the main function of the stalk is not in the distance of these couple of millimeters or fractions of a millimeter only. The spores will be elevated and have a starting point. I think the main function of the stalk is simply to poke out, to protrude the water film, which is covering that substrate with Plasmodia and amoebae, to get the spores airborne by drying them out. The spores have first to be dry, and then they get airborne. Once they are dry, as we had seen with research of my colleague Jan Wojciechowski, the spores then are no longer around. They look like a kind of football, you know, American football, these kind of spindle-shaped things, since they lose a little bit water. And now, coming back to citizen science, we have photographers among them, which can show that with direct stack macrophotography. It's just amazing to me. So the main thing is get the spores dry, and then you can occupy habitat islands with a particular high density of prey of other microorganisms. And that is the point, and that is the huge evolutionary advantage of spore formation, and that's in my eyes the explanation why spore formation occurred several times parallel in living organisms. And here's my example showing the difficulties. Where's the stalk? The stalk is gone in these Dihyopsis racinae. Here you see the fructifications first developing on a very unique habitat. It's stem wounds of coniferous trees overgrown with resin exudates, and on this resin, a fungal bacterial community, we will come back to it, develops. And as one of the predators of this community, these Dihyopsis racinae comes, but where's the stalk? The stalk, of course, which is so advantageous in evolution, immediately turns into a burden if you can rely on substrata which regularly dry out. If they regularly dry out, you're simply going to wait until all is dry, and then you can disperse your spores without the stalk. And as you will see on these photographs A, B, C, you see that the spores can dry out since the bark of the tree, and these photographs is made basically at breast height. So you have a spruce tree and at breast height, in its stem wounds, these things fruit. Yeah, of course, eventually they dry out, and then the spores can become airborne. And of course, that is a nice example how a structure can get quickly lost if the particular microhabitat allows it. Perhaps this thing has a special trophic ecology. We will see it has a pronounced genetic structure. It is probably another species complex with unresolved phylogeny, and highlighting again the role of citizen science. It was discovered by my colleagues Bernhard Wörli and Henrik Götzsche, both non-institutional researchers, as I want to call them. They were the first to spot it, since they had the endurance and the idea to look at unusual kinds, at unusual substrates. Yeah, and then the morphology. Not very rewarding, I would say. Pretty nondescript. Black spores, it's a member of the dark spore tribe. Descript capellitium. Of course, as you see here under F, the peridium is translucent, and the colors, these wonderful silvery blue iridescent colors, are false colors. And since the color is determined by the diameter of the peridium, which is in the range of the visible light, so between 400 and 700 nanometers, slight changes, environmental cause probably, in peridium thickness will cause dramatic changes in the false colors. So sometimes they look like C, a little bit dull gray, when the peridium is so thick that it's out of the range of the visible light. If it's not drying out properly. If it's drying out properly, the true peridium, and that's then rid of the slime layer, is in the range of the visible light, and then these false colors, like in hummingbirds and so on, will appear. But here the false colors will be done, I don't know if you can write here, will be done by interference of the light reflected from the lower and the upper margin of the peridium. So a pretty nondescript morphology. We need more than morphology. That's the point here. Yeah, when we look at Myxomycete diversity, we are of course tempted always to look at a regularly fruiting species. That's our profession, and looking at the fruiting bodies has been, as we know from this nice paper of Steve Stevenson in 2008, for 200 years, the profession of mycologists. And so the Myxomycetes are one of the few protist groups which can look back on a history of research for more than 200 years. The nomenclatural starting point is even earlier than that of the true fungi, with Linnaeus species plantarum in 1753, but there are more. Molecular investigations, and I will tell you the idea once more, since we need that for our example species, have been showing that we have numerous cases of cryptic speciation, and metabarcoding has given us a little glimpse into the hidden dimension of Myxomycete diversity. And I would even speculate, here we can do it among us in a scientific paper. I would be, of course, a little bit more reluctant with this, but I can speculate maybe Myxomycetes can lose their ability to fruit. My colleague, Oleg Shepin, will explain that a little bit more in detail in his talk. So if you look at these very familiar Myxomycete life cycle, I'm reluctant to call that an achievement. Of course it is, but it goes back to Debarry 1863, if I'm right. So it's very, very long known. Most of this Myxomycete life cycle is in the dark. When we do field research, this is for sure for us. And most often, even the Plasmodia on Strocia we don't see. So all what remains from this very interesting life cycle are the trophic structures of the fruit bodies. Of course, this is our main focus of research, but we should always try to look in the other stuff as well, even if it is super difficult. And here you see a little, let's say, summary of the taxonomic research on Myxomycetes. The red line is the number of described species. And you see the blue line, sorry, I was wrong. The blue line is the number of described species, and you see this number ends at whatever 1,700 or so. Of course, numerous were put in the synonymy. In the second round, I will extract those species which are nowadays recognized with the help of the famous database of my colleague Carlos Lado. And these are a few more than 1,000. So that would be then the real curve. But you see, after slow progress, interrupted by a few jumps by the major monographies we all know, and here I have used the author abbreviations as we use them in the Myxomycete names when we are giving authorities. Since, let me have a closer look. Since about the 60s, roughly the end of the 60s, we have a steady increase in new Myxomycete species. And a little bit lagging behind is the red curve, the number of new combinations, which reflects changes in the system of the Myxomycetes. And if I should have a projection, I think it's especially the red curve, which will see a steep increase within the next number of decades, maybe even within the next decade, since right now we have a full number of interesting molecular papers and works, and there will be more to follow, which will just cause a kind of explosion of these new combinations. I will go back to that. And of course, even with the traditional visible dimension of diversity, there's still lots of things to discover. In spring, I was visiting Australia, and my Australian colleagues told me always, we have difficulties with your determination books and your descriptions. It all doesn't fit really, and so on and so on. So we systematically sampled staff for barcoding, and it's not yet published, but I can tell you, of course, the barcodes are different. In this region here, for example, which we had visited, it's southeastern Australia, New Zealand, and Tasmania, we have our own little temperate zone, which has also super interesting plants, but the time is too short to tell you about this. And it has you can guess different Myxomycete species like this one, which is right now under description, and the name chosen for it is Tasmania Myxa umbilicata. It's a strange Lamproderma-like thing, which has an intermediate position between probably Pipiniaceae and Lamprodermataceae. You see that on the capelitium. It's a real strange thing. In difference to Lamproderma, where you have this kind of little tree left when the peridium opens, here the capelitium, as you see in the last little and the O, here this. The capelitium will go with the peridium a little bit like when the derma is opening. Really strange thing. The thing is, if you know the habitats, as you see the red dots, this is the known distribution, not super uncommon. And most of these red dots, I would say all, are from the non-institutional researchers working here with very nice means of photography. We were not at the right time. I could see only the remnants, but even the remnants were quite distinctive. So there's more to discover if we look systematically also in more remote parts of that planet. That's one point of the visible dimension of diversity. Another point, for example, is if we do a systematic survey in areas where we usually think we know the Myxomycetes. So these are the results of a survey for the nidicolous species of Diderma. Diderma, okay, it's a large group of Myxomycetes, you know. The nidicolous species within Diderma, this is now clear, form not a monophyletic group, but again a guild. A guild means non-related organisms all together by adopting a common lifestyle. And that's exactly what is the case with the nidicolous Didermas. We have 6.5 species, so to say. With molecular methods, you can split Diderma alpinum in Diderma alpinum 1 and Diderma alpinum 2. And the Diderma alpinum 2, that is the blue, light blue group, and I hope I can show you that. I always have difficulties with my dot here. That is so far found mostly in the Spanish Sierra Nevada, which is among the European mountain ranges, one of the most deviating when it comes to the nidicolous myxes. So my colleague Gabriel Moreno is right in describing quite a number of Myxomycetes in recent years from that little mountain range, which is not larger than the high Tatra Mountains on the boundary Poland to Czechia. And you see here the results from systematically molecular investigation via barcoding from 800 species. Just by coincidence, the number was around 800. 800 species from 8 mountain ranges, and you see, and I have written that on the slide, they are differentially distributed, which means, for sure, in Myxomycetes, the thing, everything is everywhere, is all worthy, is not true anymore. Yes, we have long distance for dispersal. Yes, it can happen, but it does not happen frequently enough to populate all the mountains with one species, and it does not happen frequently enough to probably maintain gene flow within isolated populations of the species. Of course, these are only the fructifications. We have no good data yet, or let me tell you more about the distribution of the amoebae. Maybe that the amoebae are a little bit more universally distributed, but that habitat ultimately selects for fruiting or non-fruiting, that only in the right habitats the thing will come to fruit. And a species like this Diderma alpinum 2 is not only in the Sierra Nevada, but it's also in the German Alps. We only never see it fruiting. So there's a lot more to do, even in areas we think we know very well for a group of myxomycetes like these Niviculus didermas, and what is a little bit ironically in the field. I have most difficulties to determine not very well developed Diderma alpine. Molecular Diderma alpine is the least diverse and the most clear-cut of the 7 species. It's really nice. But I fail most often with Diderma alpinum. And what may help to get out these things are species distribution models. Again, another achievement of science. We have now these ready-to-go species distribution models. If we have enough data, again, the non-institutional researchers play a very, very important role in recording these data in web pages like the Facebook Slime Mold Appreciation Group, like GBIF, and so on. We can run these species distribution models. The ecologists have introduced the abbreviation SDM for that. And you see here such a model in Physum albescens, which is a kind of model species for Niviculus myxus. And you see an incidence map of the world. Oops, sorry, I was too fast. You see an incidence map of the world, and these 3 areas, which I have enlarged in these bigger boxes there, ABC, these 3 areas seem to be the hotspots for Niviculus myxomycetes in the world. But who wants to go to the Himalayan mountains? Oh, I always hear the Himalayan mountains. The model brings them out. That's fine, but probably the model will react different if you have quite a number of records from the Himalayan mountains. So, unexplored areas. Interesting is in the upper right corner, this relationship between northern latitude and elevation, where you find the records. The map is compiled from 537 independent places where we found Physum albescens, so numerous collections from one place where counted only as one, so 500 and so on, independent places. And you see these nice dependency, what I have here extrapolated with this regression curve, and you see very nicely. So the further north you go, the lower you find alpine myxomycete sitting, we know from experience as well. And it seems to be difficult to find anything more southwards than 20 degrees. I had a friend climbing the Kilimanjaro, and I had him explain, look, and so on and so on. No, he wasn't successful. He showed me photographs, and I was also thinking, okay, Kilimanjaro should be not on the urgent to-do list. And that is the second dimension, diversity. What I like to call cryptic diversity or cryptic speciation. And we have done a number of case studies, and you see them listed here in the lower left corner of the slide. When you look with molecular markers, more specifically with a combination of 2 independently inherited molecular markers to that story, you see that you have pronounced groups, like in this 3-gene phylogeny of Physum albescens, where you have these groups underlaid in different colors. The particular color doesn't matter. I only wanted to have the colors to tell the groups apart from each other. And on the right margin of the slide, you see the combinations between these independent markers. In this case, these are true. A mitochondrial gene called COX-1, the well-known 18S rDNA called SSU, small subunit of the ribosomal DNA, and the truly single copy nuclear gene elongation factor 1 alpha, which is common to all eukaryotes. You see that they form more or less exclusive groups. So the recombination takes place always within a group, as you all, as you see, for example, here, and here, and so on. Usually it takes place within a group. Other exceptions prove the rule. We have here in the more closely related groups we call HB and HA. These numbers are arbitrarily, of course, as well. Here we have a few cases of intra-group or in-between group recombination. And if you do a statistical test, of course, you will end with a super high significance that the recombination, when you would have Myxomycete, which means every amoeba can make it, every other amoeba, you will come out with a completely different picture, and it would all be intermingled. So you have local, more or less local or regional populations which don't like each other so much, to say it that way. So, for example, the group HB is probably a northern group, since KN stands for Kamchatka and KHB stands for the Kagin Mountains, the Kivini Mountains, which is a mountain range on the Kola Peninsula in the White Sea. Whereas other groups are probably more southern. The group G, that is the light blue group, stands for SN, Sierra Nevada, and CAU, Caucasus. These are more southern mountain ranges. That is one of the problems. Disentangle geographic isolation from real reproductive isolation, which may be caused by yet unknown molecular mechanisms. And that's our challenge here with these cryptic speciation. I think we have two challenges. The first is, can we find morphological correlates to the cryptic groups? We have not succeeded so far in the first of these studies. And the first of these studies was on the Myxomycete ornament, Trichia varia, described by a good old person from 1794, if I'm right. And maybe person had a feeling. Since he called the organism varia, the variant. And yes, indeed, Trichia varia in Europe is made of 3 cryptic species, which are molecularly super distinctive. The first barcoding will tell you, but the 3 species have been found near Greifswald when we did a study altogether on one. So we do not find regional differences, we do not find habitat differences, at least not easily visible habitat differences. That is the importance of the prey. Maybe species 1 has another prey than species 2, but nobody knows. And we have not been successful to find morphological differences between these 3 species. You look at them, can't tell anything. You do barcoding, you know immediately which of the 3 cryptic species your specimens belong to. And that is the challenge, but also the challenge to disentangle the underlying biological mechanisms. The hypothesis 1 might be that we have insufficient dispersal capacity, so the things stay where they are. Obviously not for Trichia varia, as I have told you, but that causes cryptic speciation by geographic isolation, a mechanism which is super well known for vascular plants, where we have speciation simply by geographic separation, since seeds of plants can't that readily disperse like spores of Myxomycetes. And the second thing is, that's only a hypothesis so far. The other way around. The dispersal capacity is still relatively high enough that in a given habitat, you always have immigrants from outside, which are not locally adapted to that habitat. If they would crossbreed with the locals, to say so, which means the local amoebal strains, they would deteriorate their local genetic adaptation. A phenomenon called by population biologists, and it's well known, it is part of the textbook, as outbreeding depression. And if you think further, a mechanism to prevent outbreeding depression would be to develop just a kind of, as I say it now, unknown molecular mechanism which is preventing the immigrants to mate with the locals. And that could be one mechanism leading to sympatric speciation, since when we look at this pattern of Myxomycete, we see a kind of mixture between geographic separation, as I have called you, but also just kind of that members of, or in one mountain, you have members of different groups side by side. Look at the abbreviation SN for Sierra Nevada. It will show up several times in this tree, or the abbreviation RON, which means Rocky Mountains, which will show up several times in different groups in this tree. So both mechanisms seem to be not mutually exclusive, and we may follow both hypotheses, but we don't know about the mechanism yet. There's a very nice talk of a Japanese colleague. I don't know who it is right now, but I'm really hopeful that we will learn a little bit more. Let's see. So the problem is right now we cannot distinguish, and therefore I've marked this slide in red as challenges we face. We cannot distinguish very nicely between this hypothetical incompatibility mechanism, which can lead to sympatric speciation if it exists at all, and the very well-known textbook knowledge commenting of allopatric speciation by geographic isolation. Simply, the spores do not fly around between the two mountain ranges, between the two forest patches often enough. And of course, the practical problem we face, and we will have a nice discussion moderated by Dimitro about this, should we describe these cryptic species? When we have these three trichias, which are molecularly so distinctive, should we describe them even if we do not find morphological correlates of the molecular differences, since we look, of course, when we do these molecular investigations, not at the genes responsible for morphology. That's a little bit our another challenge. We look at completely different genes, which have usually no morphological expression. We need to get a hold of these genes which express the actual characters, if there is a storm, how the spore ornamentation will develop, and so on. I will come back to that. Then, of course, we will see, okay, we might find a little bit of a better explanation, but should we describe such species or not? So far, I have been choosing the conservative way and say, better not. And the third dimension is the hidden dimension of species diversity. Different fruiting propensities in different regions give a hint to that. What you see here is a transect where we did systematic moist chamber studies 2020. After the last Congress, we went to the mountains of Costa Rica and did this transect. It was simply a moist chamber approach with a kind of nested design, 5 plots, each plots with 5 subplots, each subplot with 12 substrate samples, and you see here the types of litter, bark, and wood we had investigated. So one data point, one plot, then at the end, if you multiply the numbers, will be equivalent to 60 moist chambers, and you see here these graphs which show a common tendency, proportion of positive moist chambers, the absolute number of sporocarps that you see in the upper right, the sporocarps per moist chamber, the average, and the diversity, the number of units, species. These are the black dots and ribotypes of dark spot Myxomycetes only. These are the gray dots per plot. And you see a common tendency. The higher you go in elevation, the lower the overall fruiting propensity of Myxomycetes becomes. A thing I know since many, many years, if you do a moist chamber in a desert region, and I think Karina can confirm it, they pop up. If you do a moist chamber in a cloud forest, big disappointment. I still remember my first trip to Costa Rica, and I sampled a ton of moist chambers in the cloud forest under a volcano, and then I was deeply disappointed having looked at empty moist chambers for several months. Maybe elevation, which is parallel to precipitation and, of course, overall moisture, and that is my hypothesis or my speculation. I would still be reluctant to call it a hypothesis. It runs that way in these permanently moist cloud forests up there. The stalk is useless. The fructification is useless. The spores will never become that dry that they can become airborne. If they can't become airborne, they won't disperse. So the whole structure is granular useless. In other words, we don't see the fructification anymore. Maybe may live happily in these forests. We need metabolic studies, but no fructification anymore. And so I think we have three dimensions of diversity: the morphospecies, the visible dimension we all know, and I have here pictured it as an iceberg, where you know that 9/10 of the iceberg are underwater. And that's a dimension we all know, the visible dimension of diversity. I always have difficulties with my red dot. Here it is. The cryptic dimension, and if this is proven to be the rule instead of the exception, it will probably multiply the number of species. Big problem, describing or not. And then comes on top, completely unknown, the deepest parts of the iceberg, this hidden dimension. Maybe we have Myxomycete species which will never fruit, or fruit so rarely that we never see them. For example, there's a thing described like Lamproderma carpathiensis. I don't know, I think it's not validly described right now. Not yet. We have one fruiting, and there is a sequence from a metabarcode study matching exactly that fruiting, and that's it. And back to our example here, you see exactly that in this thing which we call the Archaeopsis resinae. In the two genes we have studied, which is SSU and EF1 alpha, you see parallel three groups here pictured with frames in blue, red, and golden. And these two frames, they just will translate to monophyletic clades in these phylogenies. And in the center of the slide, you see the common thing, the recombination pattern, that you have recombination within a group, but not in between the groups. So you can do quite nice things, especially with EF1 alpha, which is a protein encoding genes. And here you see a long and boring table about the variable position in this gene. And if you look at the reading frame, which you see in these numbers here, you see that the number 3 is more common than all other numbers, as you can expect, since it's the third position of an amino acid with the reading frame, which causes a synonymous mutation, which means the amino acid will still be the same to do the degeneration of the genetic code. And that will help, for example, also to recover these groups from EF1 alpha alone. Here you see a very interesting graph, which is a network of the hypothetical mutational steps for all the EF1 alpha sequences we have disentangled. So the straight lines are the network, and the arches are the allelic combination of a particular specimen. If you have a dotted arch, you have only one specimen of that kind. If you have other lines like this thick line you see up here, which is the most common allele, you have here a very thick line like a handle. That means numerous species show that combination. And what you see, okay, the three groups again will emerge. Although if you look here, if you look very attentively, you see again one case of intra-group recombination. It's SSU1 with EF1 alpha 1. The numbers, just by coincidence, we have simply given numbers. Which means, okay, we have here again cryptic speciation, and again we seem to fail with looking at the morphological correlates. Here you see wonderful SEM pictures of the Spanish group around Gabriel Moreno, just with the spores. They look very nice, but yeah, you see the colored frames depicting the groups. It's hard to spot real nice differences. And also, if you look at the distribution of our new species, which was found in different regions of Europe, you see that in most of the region, or in the 2 regions where we have 2 from the 3 regions, we have all 3 groups. Only in Denmark, the rarest group, Res2b, I call them tentatively Resine group 2b, seems to be absent. That might simply by coincidence, since the golden group is also the rarest group. So the point is, and that's the challenge, integrative taxonomy, we must get all methods we can to have taxonomic research beyond the oval characters we always have. And even the characters we always look at first might be misleading, as this study of my colleague Jan Wojciechowski is showing. Here you see 6 specimens of Physarum albescens measured quantitatively for spores. Quantitatively means here that you measure several thousand spores, as you see here at that line. This is the average spore count. Several thousand spores with image processing. So automatically go. And you see very nice graphs in spore variation. These are the box plots. And in one of the species, you even see a second spore population. If I could find my red dot, but you see it a little bit shaded, grayed out, the second spore population. Here you go. Which is probably multinucleate. You have to read the paper. And now comes the point. A study employing a method called genotyping by sequence, which is the blue genotyping method, has proven that all 6 specimens belong to one and the same clone cell in 2007. Which means this is environmental induced variation in spore size at the very end. So even such a character, don't look at spore differences of 1 micrometer, and so it is not a reliable character. That's a bad news to distinguish between species. But you have other characters, as this wonderful study of Dimitro has been shown. He is working with Leucogala, and you all know the boring Leucogala evidently. You never sample, please sample every day, especially the smaller ones. In our herbarium, Dimitro has piled up the boxes. And when I came down 2 days ago, he had numbered the piles, and he is now at number, what was it, 76 or something? 76 of the piles. This shiny Leucogala, you have an every second long, is a huge cryptic species complex, and Dimitro managed here with these, you see it at this nice color plate from a recent publication, he managed to find a suite of new characters, especially laying in the peridium. I don't want to go into details from time reasons, which distinguish species. So we could find morphological correlates to the cryptic speciation. The quest is not useless. You can have success, but don't count on it. And you see also the variation in spore patterns. This is staining with, what was it, methyl violet, right? Methyl blue, and you see the huge variation. More color plates in this wonderful paper. But coming back to our example species, Dihyopsis resine, here the quantitative spore measurement was also not successful. No really significant differences between the blue, red, and the golden groups. As you see here, from a number of specimens, we have quantitatively And that is the problem. We have critically to ask for the phylogenetic value of the nicely visible characters. And unfortunately, this is how biology plays. The most eye-catching characters may be those which are under highest evolutionary pressure, which are highest selected for, and these are those which are most variable if you look at groups of species. And that's a little bit of a problem. And the rule number one I would here propose from an example you all know, for example, Didymium versus Mucilago. Mucilago is no more an own group, an own genus, since molecularly it's clearly a Didymium. Mucilago is embedded in a clade of solitary fruiting Didymiums, usually even stalked. And so it had to be recombined to Didymium. We first say Didymium mucilago, but Carlos picked up a rule which is forbidding this name. So right now it's called Didymium spongiosum, according to the basionym, if I'm right. So compound fructification, the same probably will happen if somebody is looking molecularly at Cribraria, which would be a rewarding task as well. Cribraria versus Lindbladia, I also think Lindbladia will not deserve generic rank anymore. So these eye-catching character, compound versus solitary fructification, probably is nice for species delimitation, but has not a phylogenetic value in higher ranks. Another thing is the spore ornamentation. And I have brought you a few examples, since I'm a botanist by professions, in the vascular plants. And especially spore ornamentation, as I will show you, is a nice character, an eye-catching character, of course, but it might not have pronounced phylogenetic value. For example, I think Irina, where is Irina just had it in her paper, Lamproderma cristatum and Lamproderma arctioides are nearly identical in outputs. And you know the strikingly different spore ornamentation of Lamproderma cristatum. I will show you that in a minute. If a species is differing by only a single character, even if it is a super easy, eye-catching character, be a little bit suspicious. It might be a species on its own, that's fine. And indeed, in the multi-gene by phylogeny, as we'll see with Angela's poster on Lamproderma, we can distinguish two species. But the striking character spore ornamentation is probably not the character which you can use to justify higher taxonomic ranks. Here you see that in an illustration, again, our new species, and I was just thinking, we all were thinking, how should we call it? Coloderma, Diachiopsis, or is it the Lamproderma which we specified? Okay, the first step, the Lamproderma, since as you all know, Lamproderma and Diachiopsis have one striking character, they differ from each other, and it's the absence character, the absence of store in Diachiopsis. And if you look at this tree, you see all what is not covered is Lamproderma, except for the outgroup on the top, that is Meriderma, we used here as outgroup, and that's a plain 18S rDNA3, and you see in between intermingled a number of other genera. Actually, the color for Coloderma does not come over properly. I don't know why. Coloderma is the pinkish color, so here it's now coming over as green. That's wrong. So think this is pinkish, please. And you see green, that is right, is Diachiopsis. And you see Diachiopsis being intermingled in Lamproderma. And the continuation of this tree shows it very nicely. And here is this group. Now I need my little dot. Here is this big group here is Lamproderma arctioides, Lamproderma cristatum, Diathiopsis metallica, and there's even one species of Diathiopsis reticulospora in it, but this might be simply misdetermined. So let's just refrain from that. But the other species are represented by numerous specimens. I don't think that they are misidentified. And you see often the numbers NM also sometimes, and Marianne Meyer, as we all know, basically knows what she's doing. So if you look at these three species, the Ecology metallica, Lamproderma cristatum, Arceoides, and pulveratum, I've got to mention these species have three stalk fruit bodies, one is sessile. And three others, have spinulospore ornamentation, and one is with these strange crystals for ornamentation. It might well be, we don't know the underlying genes, it might well be that a single gene or two genes may cause a difference in this eye-catching character as we know it from vascular plants. And I have plundered our botanical garden and brought you little herbarium sheets of vascular plants. You can have a look at them. And these little herbarium sheets show a laciate version. You probably know it very well. They show a laciate version of the plant versus a non-laciate version of the very same species. In vascular plants, of course, it's very, very well known that you have this species, and the students used to challenge me in showing me a twig of a Laxinian alga, and then I have a very hard time to see what it is. But since plants have a plethora of visible characters, I usually can pass the exam. With myxos, it might be more difficult. The mechanism leading the lacinate leaves, I can show you later a slide, is right now known from our developers Thaliana, but the scientists have found a model. It's overexpression in the single gene, which is causing lacinate leaves. So that might also happen in spores in Myxomycetes. And this is the last combination of the three. This is our new species, which is not entirely monophyletic group according to the barcoding marker. Of course, the barcoding marker is not good for phylogeny, for ecophylogeny at least. So you need two markers at least to deliver it as a monophyletic unit. Okay, Myxomycete systematics and the quest for the natural system. That is nearly my last point. And that is a point we also have all problems with it. It's fascinating, but we are right now in a position where we have the tools, where we shattered the traditional system basically in its foundation by applying these tools. But our new knowledge coming from the molecular results is not yet comprehensive enough to erect a new system. Basically, we have demolished our building and in the long-term process to build a new house for our Myxomycetes. And of course, all these new combinations will cause instability of names. And I think now you understand why. Finding morphological correlates, the limiting molecular groups, is really, really difficult. Here is a very rough blueprint of a new house, but it's very rough since it's only relying on entire 18S rDNA sequences. It might be partially wrong, although more of it was confirmed by recent multi-gene phylogenies. And here you see an overview of the literature, but the results are difficult to interpret. The reason is delivered here in this schematic picture of this paper, which shows the expression of characters in different groups of Myxomycetes. Characters can frequently go lost. Then they are reinvented, slightly different, but they are still reinvented. And all these double blocks here, like this one, for example, this is the loss of a character, which quite often appears, and a primary stalk, which is early lost in the evolution of the bright spot, is, as you then know, replaced by another type of stalk, the supravoltaic, which is also a stalk, also serving its function, but newly invented. And these are our challenges. Parallel evolution of structures is the rule, not the exception in Myxomycetes. And losses and reinvention of structures are frequently going on. A good example for the parallel evolution of structures is the compound fructification. A good example for loss and reinvention of structures is the stalk. So our challenges are 1, 2, 3. One is we need to use genomic methods, and that's difficult for non-cultivated organisms to just have stable phylogenies. Genome skimming might be a point. We need to find exclusive characters for a group, which is a real challenge due to this frequent loss and reinvention and parallel evolution. And we need, at least that's my opinion, to perform the transition to a natural system with the least number of new combinations. So probably we have frequently to ask the nomenclatural committee to conserve names. Here you see an example of the latest paper of the LATO group. They have done a wonderful study in Persunia on Physarum. And here you see one group of Physarum, which these authors have recombined into a new genus, Nanengella. You know the etymology of the name instantly, of course. But look at the species. In this group, which is a well-supported monophyletic unit, you find compound and solitary fruit bodies, stalked and sessile fruit bodies, single and double parencia. The columella, pseudocolumella, of course, might be present or absent. The capellitium might be physaroid or bethamuoid. The spores might be clustered or solitary, and also the lifestyle doesn't help. Tropical species are together with melliferous species. So that's a real challenge. How can you find unifying characters? And for field biologists and ecologists, these are not really good news, but this is like biologists. These are the problems we face in future. Here, for our species, to come back to our example, okay, we managed with 2 genes on the very end to find a monophyletic unit. I don't know, it's split into 4, but one is no supported, and these Res1b are exactly these 2 species which have the intergroup recombination. Understandable, of course. So, okay, but you see again in these 2 gene phylogeny, Lamproderma occurs several times. The Physaracea and the Didymyaceae come out as embedded in Lamproderma, since on top of the whole thing you have Lamproderma cacographicum. If you look thoroughly, here is Lamproderma cacographicum. And of course, what is always nice is to have species represented. These are the triangles, which are sometimes long and sometimes short, depicting intraspecific variation by several specimens. Okay, we at least got out that we deal with a new biological entity, but what are the real relationships? Angela will tell you a little bit more, but this is a real challenge. And as the last thing, the Myxomycete ecology, and here I would say our real challenge is to establish the trophic relationships. I can't show you a lot of achievement. We have tons of interesting study with proxy factors. Mao Inoue will show you a very interesting study, which is interesting at that point that we have tried to augment it with molecular methods. But when it comes to the food organisms, we have only, I would say, anecdotal results. And you see a few literatures these here mentioned. Our challenge is that right now we can only look at the toxic factors, the vegetative decline that Myxomycete. But this is not mattering a lot, since a microhabitat, which means a very small section of the substrate plus the microbiome, might be the goal for the anaerobic population. So a mountain meadow beneath a tree may be completely different from the mountain meadow one step beside, which is out of the shade of that particular tree. We also look too much to only the fruit bodies, sorry for the spelling error, and we still rarely take cryptic speciation into account, since that requires molecular measures, but it's exactly a point for cryptic speciation. It might well be speculation again that the cryptic species differ in their food preferences, and that's their real difference. A difference we can't see, of course. So that is, in my eyes, also the challenge in the whole thing. And here you see that we have several approaches. One is metabarcoding, more in Oleg's talk. One is detecting endosymbiotic bacteria. Another one is culture feeding trials. I think Johan will tell us a little bit about this in his talk, at least being able to quantify amoebal populations in the soil. But I think this is a field where we need to make progress, and it's damn hard to do, since as you see on this small list, all these approaches have their shortcuts. And that's a real problem. Back to our species, that science, apart from the problem that we cannot have a right name for it, apart from the problem that we do not have very distinctive morphological characters, this seems to be a promising species with its very, very special microhabitat. Since here you see the stem wounds, these are photographs from the Black Forest for my colleague Flavius Popa, who is the mycologist in the Black Forest National Park, a very good mycologist. And now we brought him into Myxomycete, and we do research together. And this is the habitats, and you see these black frame around the stem bounds. Here, look, this is where the species appears, in 1 to 2 meters high on the trees. And that very special habitat would give us a chance to detect the food organisms. If you come and see, and the phenology of the species is a strange one. The fructification peak, at least here, seems to be in February. In February, you sit on your desk and write papers and do not search for Myxomycete in the field, you know. You're out. You ski. You have to and look at the stem bounds. So we tried, of course, with metabarcoding, and metabarcoding indeed gave interesting results. I will not dive here in the method, since Oleg can tell you a lot more than I can do about this. But metabarcoding, a kind of barcoding of communities using next generation sequencing, can reveal, if you have the right markers, the organisms of the whole community. And here we tried with different markers, of course, to look for Ascomycetes and for bacteria. Yeah, usually the results are a little bit boring. You have numbers, so I have visualized the results for you in these two graphs. The first one in the upper right is the graph for bacteria. The second one is the graph for fungi. And you see, of course, pronounced differences, especially for the bacteria. Two genera, Sphingomonas and Endobacter, are the genera which are really, really abundant. So we can at least now come up with a hypothesis. Okay, maybe these are the food. We don't know. It's only a hypothesis. It's only association by maybe these are the right food organisms, and these Ascomycetes are probably paving the food for the new community, since these Ascomycetes are able to degrade a natural polymer, a kind of natural plastic, which we know as resin of the trees. These fungi can do it, and you can use them as well readily as indicator organisms. And here you see the fruitings of some of these organisms, quite interesting. These are all little Ascomycetes, only recognized for specialists, but okay, we are well acquainted with the range of these Alopecia. It's not a big problem to find them for us, I think, if you know how they look like. And there's a ton of descriptions out. So these Variatiformis with very inconspicuous red black apothecia, these super small apothecia of less than a millimeter here, is one of the best indicator organisms for the presence of the Myxomycetes, whose fruitings are a lot more elusive, whose fruitings weather a lot faster. Usually your best thing is the black crust. And this is a nice thing where we should also pay attention, the microhabitat. Looking at the microhabitat, since this is a way, in my eyes, how we can bring Myxomycetes also into nature conservation, telling the conservationists, look, these stem wounds, this is not the thing you should try to avoid. It's not a bad thing. It's, again, adding to the diversity in your national park, and that's exactly the message we have conveyed. So to summarize the whole thing, since now I'm on the end, to summarize the new thing, we have a number of challenges. We need, I think, to look more frequently, a typo again, we need to look more frequently to other groups of protists. These guys working with other groups of protists doing nice and amazing things, and often they are better funded than we. One of the best studied groups is Enteroamoeba. And when I did a journey in 2009 to Madagascar, I learned why, since Enteroamoeba found me. And I suffered a huge trip from Enteroamoeba. So there's a lot of money going into research of this medical relevant organism, which is relatively close, to our organism, not forming fructifications, unfortunately. So it might be interesting to look at other groups, what they are doing. That's the first thing. The second thing is, let's explore the cryptic and hidden dimension of Myxomycete diversity. It's difficult, but it's fascinating, and we will come up probably with a lot more and interesting results. Let's look around what methods people are using for taxonomy, to come to an integrative taxonomy, to find also new methods, like, for example, these mass counting quantitative analysis of spores, what I have mentioned to you, or look in ecology also with new methods are things like aerial spore dispersal. Jan will tell you something about that. Let's try to look to the food organisms of the Myxomycetes to establish real trophic relationships. I think that is a very difficult thing, but really as a challenge we need. And of course, last but not least, let's cope with the many nomenclatural changes we will probably face in the next decades when we do the difficult transition to a natural system in Myxomycetes. There will be a discussion again, good taxonomic practice. I think the challenge will be to find the right balance between doing a new combination and saying, okay, let's be modest, let's wait for more results before we do a new combination. So, many thanks for listening to this, I know, rather comprehensive talk, which has just pointed to a number of fields, but I think as an entry for our Congress is just suited right. I'm grateful to a number of individuals, and this is really important. But still, I want to emphasize this is the nice thing, and this is what I have always enjoyed in my life, working together with so many different people from different countries, from different origins and cultures. This was really nice, and I think this is also a justification also in these hard times, to go on with science, connect with people. That is what we need. Thanks a lot for attention. I'm ready to spread the talk, and there's also a literature list at the end of these slides, which I will not show you. I'm ready to spread the talk if you wish. That's the PDF. Thank you.
The fantastic 10 minutes for questions. Yes.
I'm glad that I can overdo. I know it's not a big difficulty because I was given a lot of time. Yeah.
Excellent talk. Thank you. I'm Nathan Yost. The Dichiopsis resina, is that been formally described yet? Yes? No? Not yet. Okay. Because when I first heard about it a couple of years ago, I went out to the outskirts of Oslo in April, and I found it on the first tree. And then we found it a few other times. I'm sure it's the same. I've sent pictures to Henrik and so on. To me, it's on the wounds on rather young spruce trees. It's colonizing or appearing on the black furry old resin, colonized by, I think, Sorosibe resina mostly. And to me, that creates a dark, furry, nice, hot place. The fur, it absorbs moisture and heat, and is a very nice environment for Plasmodia to thrive. So have you emphasized the ecology will you be emphasizing the ecology when you describe the new species? Yeah. Okay. The Norwegian specimens are not that rich as the ones you showed, but there are a few sporocarps there, so it would be nice. Okay.
So in the Jupiter parts of Norway, probably no problem to discover it. I went around Bergen in summer last year, and we were there in Spain. But the locals told me, "Oh, you are too late. Our summer is over. We had a very, very long dry spell of several weeks. So of course I checked the stem once, but expectedly I couldn't find anything after a very long spell of drought, and I was completely on time."
Martin, we know for a long time this hypothesis about Myxomycetes, which lost the fructification ability. But it's still a hypothesis. So how do you imagine the experiment or the data which can finally prove that such lineages exist?
I see one way. One way would be to look at the genes responsible for fructification in dark sport. We look at the chunks. Since the group of Marwan in Magdeburg they are working, they work on the genes which are responsible for protein, of course, the sound. But it's evident that when we look at these genes and they detect mutations which render the gene non-functional, especially in coding genes, it's a serious work. But that would be the way I could imagine. We could maybe prove that these genes are now dysfunctional. And then we know what it is. And of course, it's very obvious when such a gene, which is in a chain forming a fruit body, becomes dysfunctional or even gets an diagnosis. I always tell the students a kind of comparison. Again, it helps to be a botanist. You all know Anemone nemorosa, the little spring flower in the woods. And we have the wood carpet in a wood called the Lichtenhain spring. And there's one clone, at least among the millions of these little flowers, which forms instead of the flowers a little plume of small green leaves. There's a mutation in the flower genes, in these high-level genes which regulate the differentiation in flowers, it's rather well known, these genes, which renders the flowers completely non-functional. They have a little push of roots. But the clone is there, and the clone is living and thriving since they still have their vegetative reproduction by our license. But this is how I imagine the non-fruiting amoeba among Myxomycete in these cloud forests to be living. You first need interesting metabiology studies which show enough significance that such sequences are there. Then we need to isolate the amoeba, and then we need to check for these genes leading to fructification. Probably the best bet is to look for canceroids, of course, for these which are common into the studies but never found.
So you expect that these lineages are apomictic?
And that is, of course, their drawback. There's not an immediate drawback, but the drawback is, like in my little anemone, this particular clone is reproductively fertilized. It can reproduce vegetatively and apomictic. I understand this is the same like vegetatively, it comes to the genetic consequence, but it's still there. Yes, that might be the point, but then it gets into more difficult. So when you have these apomictic things, then you have a lot of local sequences and race and so on, since they can't exchange information with each other. And again, I have a very nice parallel evolution example from vascular plants. Look at the dandelions, the brambles of Rufus hirasu. These are classical apomictic genera in vascular plants, and they are just doing an evolutionary relation into a lot of species which are just distinguishable by small characters, because local in Myxomycete plants show many more characters, but at least we have a limited specification. But that might be the point. We now plan a project to investigate that in the tropics. That might be the point why these tropical mountains are not so super rich.
And since these are apomictic lineages, the question arises, can we consider them as species or not? And this is especially challenging because here we have the rough analogy with a case of transmissional tumors in animals, where the multicellular organism produces a unicellular lineage, which accumulates enough mutations to be considered as another species. And theoretically, we can consider within one genus the mollusk or the marsupial mammal and the unicellular parasitic organism. And here in Myxomycetes, we have the similar macroscopic plasmodial and fruiting body forming organism and its unicellular relative.
Here you see the challenge. The legal technologies a lot of challenges, especially with Myxomycetes. There is not that of this.
Can you now send a message that everything is ready so we can do the comment break?
This is the Lazio chest.